Hello, boss. Today we want to present to you CRISPR, a technology that revolutionized the medical industry. While it can solve various diseases, we want to focus on Alzheimer's today.
Alzheimer's disease is a brain disorder that slowly destroys memory and thinking skills and eventually the skills to do a simple task. Alzheimer's disease is currently ranked as the 7th leading cause of death in the United States. Moreover, some research from the National Vital Statistics mortality in 2000 and 2010 shows a drastically increasing death rates of Alzheimer's patients for all three groups. However, the most higher death rates come from patients who are older than 85 years old. Okay, here is the diagram that shows some conditions that contribute to Alzheimer's disease to show how these diseases work. The first is cholinergic insufficiency. This leads to acetylcholine-producing neuron death, which causes to memory loss. The second one is excitotoxicity. This is when there is too much glutamate that overstimulates neurons, leading to cognitive dysfunction. The third is the amyloid plaque. This contributes to A-beta protein clumps that form outside neurons and prevent cells from signaling to each other.
See, boss, this third problem is what we want to focus on with CRISPR. This problem is caused by a mutation in the APP gene that changes certain codons. For instance, the sweetest mutation is caused by substitution of lysine and methionine to asparagine and leucine. However, with CRISPR, we can edit the gene to suppress these mutations. This is a diagram of how CRISPR works, and it can be broken down into a couple of steps. The first step is to fuse cRNA and tracer RNA duplex to form a single guide RNA called sgRNA, and this is complementary to the target site. This acts as a guide so that the Cas9 enzyme knows where to cut the DNA. Step 2 is to actually use this sgRNA to guide Cas9 to a target site where it will then perform a double-stranded break. Once this happens, the cell will repair itself in two different ways. The first way is non-homologous end joining, and this causes a frameshift mutation through an insertion or deletion mutation. The second way is HDR, homology-directed repair, which introduces a precise gene modification and is provided by a DNA template. Through this, we can suppress the Alzheimer's disease in the APP gene. However, boss, there are some other treatment methods that we want to discuss with you, specifically oral medication, which uses rivastigmine and donepezil and are popular for solving Alzheimer's disease. However, despite showing symptoms of improved cognitive functions, there are some side effects that come along with it, such as weight loss, nausea, diarrhea, and vomiting. Because of this, I don't think this is the most optimal solution for Alzheimer's disease. And looking at CRISPR, it just shows how much new technology can help us in the long run, yet there are some risks with this that we have to consider. For instance, CRISPR can lead to the off-target effect, where Cas9 accidentally cuts an unintended site, leading to mutations that we did not intend for the patient. Despite this, scientists have found solutions around this, and they have various solutions, but we want to focus on two, which is base editing and prime editing. Base editing allows for single base corrections without breaking the actual DNA strand, while prime editing allows for base substitutions, insertions, and deletions. Both of these methods are currently being experimented on, and we hope that it can lead to safer and more efficient methods for the patient. And we hope these alternatives appeal to you, boss, as these methods are revolutionary for future treatments.
This is one of the most crucial diseases which lead to death worldwide. CRISPR is one of the best options to treat disease by selectively editing the gene that related and associated to this disease. Although there are some risks of CRISPR treatment, more funding and investment is necessary to more research and development of the CRISPR treatment.